Okay. Good evening, everyone.
Good evening, sir.
Good evening, sir.
Thank you. Today we are going to discuss gametogenesis, which includes spermatogenesis as well as oogenesis. So, coming to gametogenesis. So, gametogenesis is nothing but simply the formation of gametes. So, formation of gametes is called as gametogenesis. We know in sexually reproducing organisms, gametes are of two types, sperms and ova, based on which gametogenesis can be either oogenesis or it can be spermatogenesis. And whether it is oogenesis or spermatogenesis, there are three common what we call stages or phases, we can call them so. So let me explain these common phases, then we'll move to the each topic separately. Coming to gametogenesis, it involves three phases. The first phase is called as proliferative phase, or we can also call it as multiplication phase. And during this phase, only mitotic divisions take place, which results in large number of mother cells of gametes. In case of spermatogenesis, we call them as spermatogonia. In case of oogenesis, we call them as oogonia. These mothers are respectively called as spermatogonia or oogonia, and we can simply call them as gametogonia. Okay, and after this, what happens is proliferative phase is followed by the second phase called as growth phase. So, in growth phase, what happens? The gametogonia, that means the cell, increases in size. The increase in size corresponds to one with respect to cytoplasmic constituent and the other with respect to nucleoplasmic constituents. And this is the phase which generally in size cycle we call it as G1 G2, where synthesis of DNA also takes place and each chromosome is represented by a pair of chromatids. Okay, thus the cell increases in size. After that, it enters the third phase, which is called as maturation phase. Maturation phase is the phase where the reduction division or meiosis takes place. Thus, when we see germinal cells or the reproductive cells are the cells where mitosis also takes place and meiosis also takes place. Whereas in somatic cells, only mitosis takes place. That is the basic difference. So, due to meiosis, what happens is the cells which increased in size, they divide to produce four cells. Because it is meiosis, there is one cell which is called as the mother cell, spermatogonium or oogonium. It increases in size during growth phase after multiplication. So there are a number of cells. They divide to form mitotically a number of mother cells during proliferation phase, and each cell increases in size during growth phase, and this undergoes meiosis 1 to produce two cells, which undergoes again meiosis 2 to produce four small sized cells. Thus, from each mother cell, we can get four different, four types, four cells. Thus, at the end of gametogenesis, there are multiple number of cells. But there is a small difference with respect to spermatogenesis and oogenesis in case of mammals, where only one worm is developing, whereas millions of sperms are developed due to this process of gametogenesis. And what the differences are, we are going to learn now. So before going to this, we should learn about something about the primordial germ cells. That means these are the germ cells from which the actual reproductive cells or germinal cells are derived. Primordial means the first, the cells, the germ cells which are first formed. So coming to the vertebrates, these are of uh, their origin is said to be of different types. We'll start with anurans. What are anurans? We know amphibia. Class amphibia is divided into extinct as well as extant classes, subclasses, which are again divided into orders. Epoda, Urodela, and Anura. So Epoda is represented by snake-like animals, Ichthyophis. Urodela, the name implies Euro means tail. So there are tailed amphibians like salamanders, and Anura. Euro means tail and means absent. So tailless amphibians like frog. So here in these tailless amphibians, that is frogs, what actually happens is the germ plasm is marked by granules near the pole of the fertilized egg. They are said to be marked by small granules. The germ plasm, not the germ cells. The germ plasm is marked by granules near the vegetal pole of the fertilized eggs. And the germ plasm, which is present near the vegetal pole of the fertilized egg, in due course of time, it divides to form a number of cells, then divides to form a number of cells. And the germ cells are found in cells near the vegetal pole because the germ plasm is present in the vegetal pole. Hence, the germ cells are found near the vegetal pole during cleavage. From there, they pass on to the endodermal floor of the primitive gut. When we are discussing yesterday the process of gastrulation, the blastula forms a two-layered gastrula. This two-layered gastrula has an outer ectoderm and inner endoderm. This we call it as this is called as archenteron, and this portion is called as roof of archenteron. Then this becomes the floor of the archenteron. Archenteron refers to primitive gut. Arc means primitive, enteron means gut cavity or gut. Hence, this is the floor. And from the vegetal pole of the egg, after cleavage, morulation, blastulation, gastrulation, they are seen on the endodermal floor of the primitive gut. Whereas, in case of urodeles, these primordial germ cells do not arise from the endoderm, rather they arise from the mesodermal cells. So the difference is in endurance, the primordial germ cells are of endodermal origin. In urodeles like salamanders, they are of mesodermal origin. And coming to the other vertebrates, reptiles and birds, primordial germ cells arise from the endoderm, again, as in the case of frogs. They travel through the bloodstream, and when they reach the vicinity of the mesodermal gonad rudiment, when we are discussing about sex determination, I told you that the gonad is in the form of a rudiment under the influence of the what we call genotype. it can either become a testis or it can become a ovary. So this we call it as gonadial rudiment. And we know the reproductive system develops from the mesoderm. Yesterday we have seen this. So it is called as a mesodermal gonad rudiment. So in reptiles, the primordial germ cells arise from the endoderm, but they travel through the bloodstream and they reach the mesodermal gonad rudiment. Thus, from there, they enter what we call it as the gonad. Okay, so decentering. Okay, next, in case of coming to the mammals, they are neither present in the endoderm nor they are mesodermal in origin, but the primordial germ cells are located in an appendage of the embryonic gut known as the yolk sac. This you have to remember. So in mammals, primordial germ cells arise from dash. They originate from yolk sac. From there, here if this is the baby, this is the mouth, and this is the alimentary canal. And from the midgut, yolk sac arises. From the hindgut, allantois arises. So these cells are present in the yolk sac. From there, they migrate to the hindgut, and from there, through the mesentery that suspends the hindgut, they enter the mesodermal gonad rudiment, which is by the side of embryonic kidney. So what you have to remember here is the primordial germ cells are present in the yolk sac. So in case of anurans, they are endodermal in origin. In case of urodales, they are mesodermal in origin. In case of reptiles and birds, they are again endodermal in origin. From there, they reach through the bloodstream to the gonadial rudiment. In case of mammals, they are located in yolk sac. So this is the basic fundamentals which you have to remember. And coming to the humans. In humans, the migration of primordial germ cells to the mesodermal gonad rudiment takes place up to sixth week of the embryonic development inside the womb of the mother, which we call it as gestation. During this time, somatic gonadal cells initiate the development of either a testis or an ovary. Why we are calling it as somatic gonadal cells? Because it is yet undifferentiated. This gonad rudiment is yet to be differentiated, and from there either it can develop into a testis or it can develop into a ovary. Once they reach the testis or the ovary, they proliferate within the gonad via what we call mitosis, which is the first phase. Most of the primordial germ cells reached the developing gonad by the 11th day after fertilization in case of mammals. During this passage, trek means passage, they have proliferated from an initial population of 10 to 100 cells to about 2500 to 5000 cells in the gonads just within one day. From 11th day to 12th day, they increase enormously in number due to continuous proliferation by mitotic divisions. Okay, and how we distinguish the primordial germ cells of the mammals from the other cells? The scientists have found that the primordial germ cells in mammals have a high concentration of the enzyme alkaline phosphatase when compared to the other primordial germ cells. Okay, and coming finally to the gonad. So primordial germ cells, whether they are endodermal in origin or mesodermal in origin or they originate in yolk sac, their ultimate destination is nothing but the gonad, either testis or ovary. And the gonad is found to have a dual origin. The somatic part is said to be derived from the mesoderm, and the germinal part from which the gametes are produced is derived from the endoderm. Okay, and coming to the gonads, testis and ovary. We already have discussed about it during hormonal control of reproduction in mammals. If you have any doubt with respect to this, please refer to the video that is posted there, where the entire structure of the male reproductive system and that of the female reproductive system are discussed. And testis is the region where spermatogenesis takes place, and ovary is the place where oogenesis takes place. In testis, spermatogenesis takes place in seminiferous tubules. In ovary, oogenesis takes place in Graafian follicles. In seminiferous tubules, when spermatogenesis takes place, millions of sperms are produced, whereas in oogenesis, inside the ovary, in Graafian follicles, when oogenesis takes place, only one ovum develops in case of human beings. So, how this takes place, we are going to discuss now. So, before going to that, uh, we have seen in the structure of the seminiferous tubule, there is an outer layer. This layer is called as germinal epithelium. Okay, from which enters the proliferation phase, divides mitotically, and produces a number of spermatogonia. They undergo growth phase, increase in size to produce spermatocytes, which undergo meiosis 1 and meiosis 2. They produce 4 spermatids that differentiate to form sperms. So, from each spermatogonium, 4 sperms are produced. And these sperms should be nourished throughout this entire path. And for that nourishment, there are long cells which are called as sustentacular cells. They were described by scientists called as Enrico Sertoli, hence they are called as Sertoli cells. And the sperms with their head, they go inside or they are attached to the Sertoli cells from which they draw their nutrients. Hence, Sertoli cells are found to be nutrient in function. And they have another function also, which we are going to discuss now. So here, the sustentacular cells, they rest on the basement membrane of the testis, and the cytoplasm of the cell extends to the lumen of the tubule, that is seminiferous tubule, and fills the narrow spaces between the spermatogonia, and that extends till the center, and where the sperms that are produced can get nourishment from them. Sertoli cells have different functions. We have seen that it is nutritive in function, that is what we have just discussed. In addition to this, it has three more functions. It is supportive, it is phagocytic, where it engulfs the waste and residual cytoplasm, and it is also secretory. So, supportive, nutritive, phagocytic, and secretory in function. So, what secretory function is? Scientists found that at about 8th or 9th week of fetal development of the fetus in human beings, they secrete a substance or hormone called as Mullerian inhibitory substance. In short, we can call it as MIS. And this is also called as anti-Mullerian hormone. And this anti-Mullerian hormone is responsible for suppressing the further development of the Mullerian duct system, which may lead to the development of the female, what we call ductal system. Okay, that we'll discuss later, but just remember this thing only. If I start discussing about what actually the Mullerian duct system is, it takes so much time. But for the time being, just understand this. When I start the entire physiology, I told you, I'll deal with each and every physiological aspect, starting from protozoa to human beings. So at that time, I will discuss about this entire origin of reproductive system, how it takes place. For the time being, just you remember that Sertoli cells secrete anti-Mullerian hormone. As the name implies, Anti means it is against. It suppresses the development of the Mullerian duct system, that favors the female reproductive system. And secondly, it secretes another hormone. In sexually mature testes, Sertoli cells secrete androgen binding protein. The name itself says it binds testosterone, that is androgen, male sex hormone, and also it binds to hydroxyl testosterone. And the third one is inhibin. It is responsible for the inhibition of the secretion of follicle stimulating hormone FSH by the pituitary gland. Sertoli cells thus secrete three hormones or substances. One is anti-Mullerian hormone, which suppresses the development of the Mullerian system so that only male reproductive system develops. And it secretes androgen binding protein, which binds to the male sex hormones. And it also secretes inhibin, which inhibits the secretion of follicle stimulating hormone by the pituitary gland. So these are the cells which are said to be present in the germinal epithelium of the seminiferous tubules in addition to the germinal cells. And now coming to the actual process of spermatogenesis. So for spermatogenesis, what we have to see is this is a testis. It is divided into a number of lobules, and each lobule has a number of seminiferous tubules. Okay, and when we draw a seminiferous tubule, this is a tube-like structure, small tubule. It shows an outer layer of cells. This outer layer of cells is called as germinal epithelium. Okay, this is present here in the seminiferous tubule, germinal epithelium. In the germinal epithelium only, there are two kinds of cells. One is germinal cells, the other is Sertoli cells. Okay, now we are going to discuss about, we have discussed about the Sertoli cells. Now we are going to discuss about the process of development of sperms or formation of sperms from the germinal cells of the germinal epithelium. Spermatogenesis is divided into two phases. One is called as spermatocytogenesis. It is nothing but the formation of spermatids from sperm mother cells. So you have to remember this. Spermatogonia are the sperm mother cells which increase in size to form primary spermatocytes that divide to form secondary spermatocytes that divide to form four spermatids. So the formation of spermatids from the spermatogonia or the sperm mother cells, up to this we call it as spermatocytogenesis. And now only spermatids are formed, or four cells are formed from each spermatogonium, not sperms. What happens now? This spermatid acquires a head, a middle piece, and a tail, and is differentiated to form a spermatogenesis. So this process is called as maturation of spermatids into spermatozoa or sperms, and this process is called as spermiogenesis or spermioteliosis. Spermateliosis, or you can also call it as spermioteliosis. So the spermatogenesis is of two types, is divided into two phases. Spermatogenesis, one is formation of spermatids from the spermatogonia, and the differentiation of spermatids into sperms is what we call it as spermiogenesis. And coming to the phases of spermatocytogenesis. So there are three phases. One is multiplication phase, which is also called as proliferation phase, where only mitotic division takes place. Second phase is called as growth phase, where the cell increases in size. And the third phase is called as maturation phase, where only meiosis takes place. This you have to keep in mind. And coming to the first phase, that is multiplication phase. And in multiplication phase, what happens? The primordial germ cells, which migrated from the yolk sac through the bloodstream into the gonad, they divide mitotically to produce sperm mother cells. These sperm mother cells are called as spermatogonia. So the first spermatogonia that are formed directly from primordial germ cells are called as type A1 spermatogonia. And these type A1 spermatogonia divide mitotically again and they increase in number to produce type A2 as well as type A1 again. Okay, so this is the type A1 which gives rise to type A1 cells as well as another type, that is type 2 cells also, spermatogonia. Why this is so? Unless otherwise the type A1 are said to be present, the process cannot be continued. For after the formation of sperms are over, after the formation of sperms is completed, we need sperms production even after that, because the production is a continuous process once you reach the puberty. So for which the A1 spermatogonia have to be maintained. That is the reason why here it forms type A2 as well as A1 spermatogonia. And the type A2 spermatogonia now divide to form A3 spermatogonia, followed by mitosis to form A4 spermatogonia, which again undergo mitotic division to produce intermediate spermatogonia, which again ultimately divide to form type B spermatogonia. Okay, so thus we come across with type A1, A2, A3, A4, and finally type B spermatogonia. See how many numbers of these cells are increasing. From A1, A2 is produced. A2 is divided to form A3. A3 is again divided to form A4. A4 is again divided to form B type of spermatogonia. Thus, enormous number of these spermatogonia are produced during multiplication or proliferation phase. This is followed by growth phase. So in growth phase, what happens? G1 as G2 takes place. It increases in volume with respect to cytoplasmic constituents. It also increases in DNA content with respect to nuclear contents. So this happens in type B spermatogonia. So during growth phase, type B spermatogonia increase in size by accumulation of nourishing material. They attain maturity and become what we call them as primary spermatocytes. So growth phase results in the production of dash. It results in the production of primary spermatocytes. So far this is spermatogonia, which is smaller in size, and now it undergoes growth because so many divisions have taken place, and after each division there will be growth phase also, and it has to increase to this extent. Why? Because it undergoes two meiotic divisions, so that the cells that is formed should not be very less in size. If this undergoes two meiotic divisions, what happens? The size for the first division half the size, and another division one-fourth the size, and this size is not maintained. In order to maintain this size, it has to increase to that size, so that when it divides, it produces the required sized cells, and these are called as primary spermatocytes that are formed during growth. And now it enters the maturation phase. Maturation phase, during this meiosis takes place, hence certain scientists called meiosis as maturation division. Okay, if you come across with at any place this term maturation division, you have to keep in mind that maturation division is nothing but meiosis. Why it is called so? Because it takes place during maturation phase. So now we have, after growth phase, we come across with primary spermatocytes. They undergo meiosis 1 to produce secondary spermatocytes, which undergo meiosis 2 to produce spermatids. So thus, from one spermatogonium, which increases to produce one primary spermatocyte, that undergoes first meiosis to produce two secondary spermatocytes that divide to form four small sized cells which are not differentiated yet, called as spermatids. This is what spermatocytogenesis is. Now we have to what we have to see? We have to see the process of differentiation of spermatid into spermatozoa, a process called as spermiogenesis or spermateleosis. So it shows a number of events. So it is marked by the following events. First one is here the nucleus shrinks. This is the spermatid with its nucleus. What happens? The nucleus shrinks by losing water, RNA, and even nucleolus. So the nucleus in sperm that is finally formed does not have RNA, it does not have nucleolus because they have already gone during the process of differentiation of spermatid to sperm et al. Even. The second one is the centrioles. We know centrioles are present near the nuclear membrane. Okay, so the centrioles lie at right angles to each other so that when spindle fibers are formed, they can move to the opposite direction during the process of this thing. Okay, and they are present at right angles to the each other. And the third thing is the distal centriole is responsible for the formation of flagellum. Okay, so there are two centrioles. Actually, they are like this previously. Now they are like this. So, towards the nucleus, one is present, that is called as proximal centriole, and the other is away from the nucleus, that is called as distal centriole. So, this portion becomes the sperm head and middle piece, and from the distal centriole, there develops the tail. Thus, the distal centriole develops into what we call flagellum. And now the fourth point is mitochondria becomes spirally twisted around the middle piece to form a two-stranded helical sheath called as nebuncon. Okay, so let me explain you this way. So this is a spermatid, okay, with its nucleus which gradually decreases in size. We know that near the nucleus, there is Golgi complex, isn't it? Between the plasma membrane and nuclear membrane, there is Golgi complex. Golgi complex forms a number of vesicles. Okay, and in addition to this, here what happens is during the differentiation of spermatid to spermatozoon, it forms into a number of vacuoles. It forms a number of vacuoles. These are nothing but vesicles, we can call them so. And these are filled with what we call the substances that are produced in the cisternae. And when you see under microscope and stain them, they appear like granules. So here, one of these or each vesicle is containing one granule-like thing, which we call it as proacrosomal granule. And all these vesicles of the Golgi complex fuse so that it forms into a large vesicle containing a large proacrosomal granule which spreads over the surface of the nucleus. So this is the nucleus above which this portion, the vesicle or the vacuole with proacrosomal granules which fuse to form acrosomal granule is present. And now it starts spreading on both sides of the nucleus between the plasma membrane and the nuclear membrane, and thus it forms into what we call it as an acrosome. Okay, so thus an acrosome is formed between the plasma membrane and the nuclear membrane. As these events are happening, the nucleus gradually shrinks in size by using water and it develops an indentation towards the other end. Away from the acrosome, it develops into an indentation. And we know that there are two centrioles which are now present right angles to each other. So one of these centrioles is present in the indentation, the other goes to the other side. Okay, and this one is called as proximal centriole and this one is called as distal centriole. Why we call them as so? one which is nearer to nucleus, one which is in proximity means near, proximity means near, which is in proximity to the nucleus is called as proximal centriole. One which is away from the nucleus is called as distal centriole. Both are at right angles to each other. As these events are happening, as the acrosome is said to be spreading, the membrane of the acrosome is spreading all over the three sides of the nucleus, the cytoplasm that is present here, the cytoplasm that is present in the between the plasma membrane and the nuclear membrane, it slowly comes down. It slowly comes down, along with which the mitochondria also come down. Okay, so these mitochondria, what they do, they are found to be spirally twisted. In case of human beings, there are about 24 mitochondria which are spirally twisted. And this portion, and as this is happening, what happens? The plasma membrane also spreads over this. Okay, and this forms into what we call it as middle piece, and this forms the head with acrosome and nucleus. And this is followed by a small neck region which is not yet distinguished. And from this, what happens is the distal centriole. This is the distal centriole. This acts as a basal body. It acts as a basal body. When you study euglena, okay, in euglena, what you study is euglena is like this. It has a basal granule, which we also call it as blepharoplast, from which the flagellum develops. And that basal granule in euglena is nothing but a centriole. And here this centriole acts as a basal body, we are calling it so. So this from this basal body, they develops what we call it as a long flagellum, and this forms the tail. Thus, the spermatid distinguishes to form into a spermatogene with a head region having an acrosome and a nucleus, and a middle piece where the mitochondria are spirally twisted, and a long tail which is derived from the distal centriole. Thus, it has three parts: head, middle piece, and tail. Between the head and middle piece, where these centrioles are present, we call this as a neck region, which may or may not be present in all the other species. And in so doing, the cytoplasm gradually comes down, and the mitochondria gradually comes down to form two-stranded helical sheath, and this is called as Nebenкern. So some question may be asked about what Nebenкern is. Nebenкern is nothing but the two-stranded helical sheath formed in the middle piece region, formed by the spirally twisted mitochondria. As these events are happening, the cytoplasm comes and it occupies a position that almost the entire cytoplasm is lost, because during growth phase it increases in size. Okay, and that cytoplasm is equally distributed between the two spermatocytes, secondary spermatocytes, which is again equally distributed between the four spermatids. Why? Because this much is said to be present. Very, let's say there are four parts are present. Now it is divided into two parts, and this is divided into one part. That means very less cytoplasm is present, and this less cytoplasm is sufficient for the carrying out the metabolic activities during the time of which the sperm reaches the ovum. If it has more amount of cytoplasm as in the primary spermatocyte, it cannot move towards the ovum. So in order to make it lighter, the cytoplasm is continuously lost, and it forms a layer around the sperm in the middle piece region, and this is called as manchete. So the cytoplasm of the spermatid is lost and forms a layer around the sperm, and this is called as manchete. And you have to remember these two names. One is Nebenkan, the other is manchete. The third thing that you have to remember is acrosome, which is derived from the Golgi apparatus. We know that Golgi cisternae give rise to lysosomes, and here the Golgi cisternae give rise to acrosome. Lysosomes are responsible for breakdown, lysis, and acrosome is also responsible for lysing, breakdown of the membrane of the ovum, and hence these are called as specialized lysosome. Acrosome is also called as specialized lysosome. And another thing that you have to remember is that the flagellum develops from the centriole. So this flagellum we call it as tail, and this tail region is distinguished again into two regions. So this is the cap in the head region comprising of acrosome, followed by nucleus, followed by mitochondrial engine room, and that is followed by a tail. And this tail is said to be surrounded by an axoneme, or a protoplasmic sheath. So tail is called as flagellum or axoneme, which is surrounded by protoplasmic sheath. So the tail region, which is having an axoneme plus protoplasmic sheath, is called as principal piece. And towards the end, there is no protoplasmic sheath, only axoneme is present, and that is called as end piece. So thus the tail is distinguished into two regions, principal piece and end piece. Principal piece is said to be having an axoneme surrounded by protoplasmic sheath. End piece is nothing but a naked axoneme which is not surrounded by any protoplasmic sheath. So if you say it is flagellum, the flagellum shows 9 plus 2 arrangement. This is what you know. Whether it is cilium or flagellum, in your intermediate you have studied that it shows 9 plus 2 arrangement. What is this 9 plus 2 arrangement? In this 9 plus 2 arrangement, there are two central fibers surrounded by 9 pairs of peripheral fibers. Isn't it? There are 9 pairs of peripheral fibers, and they form into these are peripheral fibers, okay, and these are the central fibers. They are present in the center, and the entire structure forms into a bundle like structure. This is what a flagellum structure is. In addition to this, here in case of sperm, there are additional additional fibers which are again 9 in number that are present outside. So these are respectively called as beta fibers, alpha fibers, and gamma fibers. So that is how all these make the tail very strong so that it can move through the reproductive tract, or in case of external fertilization in water, so that it can reach the ovum. So let's see what this structure is actually. In human beings, the total time that is required for spermatogonia to form spermatogen is said to be exactly 64 days. In some books it is written as 74, but you go for 64 days. Okay. And coming to the continuation of spermiogenesis at genetic level, gene transcription during spermatogenesis takes place during the diploid stage of meiotic prophase. Okay, we know that the meiosis during gametogenesis, meiosis is stopped at two places. One is metaphase, the other is diploid stage of meiotic prophase. And during this phase, during this block, meiotic block, at the time of diploid in prophase 1 of meiosis 1, gene transcription takes place. That is what scientists discovered is. In addition to this, the Y-specific RNA made during meiotic prophase is utilized later during the process of spermiogenesis. And scientists have also observed another phenomenon called as haploid gene expression. So this is the gene transcription after meiosis that takes place in spermatids is called as haploid gene expression is. Generally we know that in diploid organisms there are a pair of chromosomes, there are a pair of genes, and both the genes are responsible for the production of a particular character through gene expression. If only one is present, we say generally that character is said to be not completely formed. But here, what the scientists say is in spermatids also gene transcription takes place. That means even in haploid stage there is expression of the gene. We call this as haploid gene expression. An example for this, they found that T12 mutation in mice. Okay, here, it is a lethal mutation in homozygous state. That means T12, T12. On one locus there is T12 gene, mutant gene, another locus also mutant gene. So in homozygous state, it is found to be lethal and the blastomeres cannot undergo compaction in the case of what we call this particular thing. And they also observed another phenomenon called as segregation distortion. What is segregation distortion? The phenomenon exhibited by T12 heterozygote mice is called as segregate distortion. This is what we have to remember. Segregation distortion means this general Mendelian segregation is not going to take place. Mendel says that in a hybrid, a heterozygote, which is tall, having a tall gene, dominant gene, small t, recessive gene, during the process of formation of gametes, they get separated and they enter separate gametes, which we call it as segregation. But that segregation is not taking place here, hence we call it as segregation distortion. It is exhibited by the same T12 heterozygote mice. In this phenomenon, what the scientist says, genotypes deviate from expected Mendelian ratios in the progeny of a cross between two varieties or species. For example, there is two varieties where there is T12 mutant and plus means normal gene. Okay, and this is female. It is crossed with a normal male where both the genes are normal genes, and it results in 1 is to 1 T12 male and normal female. That is what general expected ratio is. In case of test cross ratio, it should be 1 is to 1. In case of monohybrid cross, it should be 3 is to 1. But the same is when it is said to be opposite, reciprocal cross. Even the male tall and female dwarf gives rise to tall only. The female tall, male dwarf gives rise to tall only. That is the general phenomenon. In a reciprocal cross, the results are found to be same. But here, when this is reversed, previously this is the female T12 plus is female. Now it is T12 plus is male. It is considered. And this is crossed with a normal female. And they found that 95% that are formed are T12 plus individuals, whether it be male or female. Thus, normal segregation or Mendelian segregation is not followed here, and this we call it as segregation distortion. Why this happens? The research is still underway. Okay, and coming to the types of sperms, there are different types of sperms. The sperms without flagella are called as non-flagellate sperms, and there are two examples. One is star shaped, the other is amoeboid. So when I have written for my first civil service preliminary examination, this question is asked: which of the following organisms show an amoeboid sperm? At the time, I did not know because I thoroughly not gone through the books. And the example that is given there is Ascaris, and another one is said to be Polyphemus. So these do not have any flagella. The second one is non-flagellate sperms. The second one is flagellate sperms. That means it is having a sperm. Examples are Amphioxus, Rana tigrina, Petromyzon, Elasmobranchs, that means Chondrichthyes, and even Homo sapiens. And there are certain sperms which have two flagella. They are called as biflagellate sperms. Example is toadfish, scientific name of which is Opsonus. So these you have to remember. I will share this slide afterwards. And coming to the functions of sperms, we know that sperms are responsible only for fertilization. But that is not just true. Their first function is to swim towards the egg because egg is stationary and only sperm has to reach the egg. So their first primary function is to swim towards the egg, and after that it has to fuse with the surface of the egg. Why it has to fuse with the surface of the egg? So that it is responsible for activating the egg. Why it has to activate the egg? We know that when I was dealing with the hormonal control of reproduction in mammals, I told you that oogonium increases in size to produce primary oocyte, which undergoes first meiotic division to produce a secondary spermatocyte and a small polar body. And if it again undergoes meiosis to ovum and another polar body is released. But this ovum has to be released during the process of ovulation. But in case of mammals, including human beings, ovulation takes place in the secondary oocyte stage only, not in the ovum stage. So once the sperm comes in contact with the what we call ovum, the ovum gets or the egg gets activated, it completes second meiosis and it becomes a haploid ovum. That is what activation here is. In addition to this, some other things also happen which we'll discuss when we deal with fertilization tomorrow. And another one is it continues to provide a haploid set of chromosomes to the zygote. These are the four main functions of the sperm. Okay, and there are certain important bits which we want to share because they are not covered in the process of spermatogenesis. There are certain enzymes associated with acrosome. They are called as acid phosphatase, cathepsin, and acrosomin. So when we deal with acrosome, we generally remember only this enzyme, hyaluronidase. But actually, hyaluronidase combined with another one called as acrosin to form an enzyme which hydrolytic in function, that is acrosomin. It is responsible for piercing through the ovum. Hence, acrosome, as it perforates or pierces through the layers of the ovum, it is called as perforatorium because it is derived from the Golgi complex and having a function of lysis like that of a lysosome. It is also called as specialized lysosome. Next, we have already seen that during the formation of what we call differentiation of spermatid, it loses water, it loses RNA and nucleolus. Thus, the sperm nucleus is not having nucleolus, RNA and fluids. Why? Because here gene expression is not required except for certain cases like haploid gene expression we have seen. And nucleolus, when nucleolus is present, it is responsible for the formation of ribosomes. Why ribosomes are required? For protein synthesis. So as there is general no gene expression, there is no protein synthesis required, there is no ribosome synthesis required, there is no requirement for nucleolus. Next, sperm nucleus contains generally nucleus, the chromosome or the chromatin is associated with histones. But here histones are replaced by another type of proteins called as protamines. These are also nucleoproteins because they are associated with the nucleic acids. And these are very arginine rich. That means a number of amino acids are present and most of the amino acids are arginine type. Hence, we call it as very arginine rich nucleoprotein. Why for this is required? This is responsible for condensation of the chromatin. So when condensation of the chromatin takes place, what actually happens? There is no gene expression. Okay, and protamines are responsible for high degree of condensation of spermatozoon, where the DNA helices are brought to the closest proximity, and it makes into what we call it as heterochromatin, which is genetically or transcriptionally inactive. Okay. In addition to this, there is an axial body. What is axial body? It is a cup-shaped depression found in the anterior face of the nucleus. Generally, we have seen that there is a depression at the posterior end where the proximal centriole is present. And they are saying that in case of many invertebrates, there is a cup-shaped depression in the anterior face of the nucleus. And this cup-shaped thing contains actin. It contains actin that gets polymerized during fertilization to form acrosomal filament that is responsible for sperm egg adhesion. So we have seen one of the functions of the sperms is to fuse with the egg surface. In case of marine invertebrates, in certain invertebrates, it is made possible by the formation of an axial body which generally contains actin in the depression present in the anterior face of the nucleus. Next one, anterior centriole and a posterior centriole. Anterior centriole is also called as proximal centriole, and when the sperm comes in contact with the ovum, it donates two things. Number one, it donates the nucleus along with 23 chromosomes. So nucleus is donated. At the same time, it also, this is the nucleus, and it has proximal centriole along with it. So as the nucleus goes in, the proximal centriole also enters. So the second thing that is donated by the sperm to the egg is the proximal centriole. What for this proximal centriole is required? When the ovum gets fertilized, it results in zygote. The zygote now enters the process of cleavage where it has to form spindle fibers during mitotic division, and the first mitotic spindle is formed by the proximal centriole during the process of cleavage after fertilization. So the significance of proximal centriole is it is donated by the sperm to the ovum during fertilization, and it helps in forming the first mitotic spindle during cleavage. Next, posterior centriole, also called as distal centriole. It acts like a basal body and forms the axonium of the sperm tail. And this sperm tail is made up of three fibers, alpha, beta, and gamma fibers. The central fibers, which are two in number, they are called as gamma fibers. And they are the site of acetylcholine esterase. Okay, they are having the activity of acetylcholine esterase. And this is surrounded by 9 pairs of fibers which are called as alpha fibers, and they are the site of ATPase. And thirdly, there are about 9, not pairs, 9 fibers which are wedge shaped present around the alpha fibers. Okay, and this is the site of ATPase, succinic dehydrogenase activity, and also a sperm lysine called as spermocin. In case of tail, there are two parts I have discussed already. Principal piece, which contains axonium plus protoplasmic sheath, and end piece, which contains axonium only. And the principal piece is found to contain all the three fibers, whereas in end piece only beta fibers and alpha fibers are said to be present. So thus from coming to the outside to inside, first gamma followed by alpha followed by beta. Okay, next, last one, fructose. So spermatozoon requires exogenous energy, food supply, and our cells depend on glucose. Mainly the brain cells. The brain cells rely only on glucose. If you give other than glucose, they won't take at all. Okay. So when you're going for the examination, you're studying day and night. Your brain gets what we call somewhat exhausted. At the time, what you have to give is just eat few biscuits or anything that has glucose so that you come back to again normalcy because brain feeds on glucose only. But sperms do not feed on glucose. Rather, they feed on fructose. You have to remember this one also. And sperm cytoplasm does not contain ribosomes. Why? Because there is no nucleolus. It does not contain endoplasmic reticulum. Why? If endoplasmic reticulum is present, if smooth ER, lipids are produced. If rough ER, it produces secretory proteins. It either produces lipids, it neither produces secretory proteins. Hence, endoplasmic reticulum is not required at all. And there is another one that is called as ring centriole, by which we get what we call distorted during the examination. Ring centriole is not at all a centriole at all. We have only two centrioles, proximal and distal centriole. Then what about this ring centriole? Here there is a sperm head followed by middle piece followed by a tail, isn't it? And there are mitochondria in the middle piece. So in order to see that mitochondria are not slipped off, they do not split off, here the membrane here in the middle piece region, the membrane is said to be highly thickened. Thus, it forms a junction-like thing. So, this fibrous thickening that forms a boundary between middle piece and tail is called as ring centriole because it is somewhat appearing like a rounded structure. If you observe it suddenly under microscope, it appears like a centriole. Hence, they called it as ring centriole. Because it is circular, it is called as annulus. Because it is entire will be set with a thickened that region is. Hence, it is called as annulus. But the correct name is it is named after the one who discovered it, Jensen. It is called as Jensen ring, and it prevents mitochondria from slipping off. And this region, the middle piece region, where the mitochondria is present, it produces the ATP. Hence, some scientists call middle piece as the engine room. This is all about spermatogenesis. Any doubts, please ask me. Otherwise, we'll move to the next topic, that is oogenesis. Shall I continue? It's already 8:50. Or we'll continue tomorrow morning.
So tomorrow morning, sir.
Okay. Any doubts up to this point? Please ask me, otherwise, we'll close here. No doubts? Can I close?
No, sir.